This doesn't make sense. Oh, it makes sense for the blood supply, but not for the skin. Hey everyone, on this episode of Coding with Kate, we are going to be deep diving into anatomy and physiology of the skin, but more importantly, cells. Now, we aren't going to be talking about the structure of cells, all of that stuff. We are going to be talking about the cell cycle, cell growth, etc., because that will help us understand how a pyogenic granuloma works. As you remember in the previous video, the vlog, where I spoke about my mother being diagnosed with having a pyogenic granuloma on her thumb and they removed it at the dermatologist, it was confirmed pyogenic granuloma. I wanted to understand how that happened, how the pyogenic granuloma functions and grows. And then once we understand that, then we can look at a sample report of how to remove it, so then we can code it in the CPT code book. Now I know we don't really need to know how pyogenic granuloma functions to code it, but just because of curiosity, I want to know, and it's actually really interesting. I was doing some research on it, some in-depth research on it specifically, and it's very fascinating. I also suggest the resources that I have are from scholarly articles, research papers, those sorts of things. So I had to do a lot of Googling what some of the terms meant that I found in there, and that is a really good way for you to learn about the topic you are learning in A&P courses. Even though you don't have to go that advanced into that knowledge, it is a great learning tool because you're actively using your mind to analyze and problem solve. And the more you do it, the more you will understand that basic information from your courses in a very in-depth way, making recall so much easier. And it's actually really exciting to go through and pinpoint, oh, I know what that means. I'm not sure what that means. I should Google it. And then you use the knowledge you've already learned to help you understand this new complex information. It's so cool. So let's get right to it because I'm really excited. Okay, so here we have my poorly drawn cell cycle chart or diagram. And the cells in our bodies, all cells, go through a cycle of growth and then mitosis, which is the actual replication or division of cells. Now, in different parts of the body or different cell types, they grow and go through the cycle at different rates. Sometimes it's very frequent, and sometimes it's long periods of them just doing their normal thing, and they don't divide, replicate, duplicate very often. So you'll find that some cells don't go through this process that much, but today with a pyogenic granuloma that's on the skin, epithelial cells do this a lot. And you can Google the exact day of turnover of epithelial cells, skin cells, but I think it's like essentially every 10 days you have new skin. That's essentially the rate it goes at, is from new skin cells being divided and formed to when they die off. That's about 10 days. I think that's the number, but it happens pretty frequently. So skin cells go through this a lot, and then also with injury, this process happens to form new skin to heal that injury, wound, etc. So let's start with our first phase. So this is our G1, this section here. So G1 is the first section of interphase. So interphase is what happens in between mitosis. We'll get to that. But within interphase, so it's like the interim in between duplication, interphase, you have G1, which is the first growth phase. G, growth. And with the growth phases, it's all, they're doing their normal thing. They're just hanging out, doing their metabolic processes. Nothing exciting is happening. They're just chilling out, being epithelial cells. G1. When that phase is complete, or that stage is complete, we get to the synthesis phase, or S1, synthesis. Synthesis is essentially the creation of something. I know that isn't the exact definition, but either way, that's how I remember it. They're beginning to create something. With synthesis, it is all DNA replication. Now, DNA replication, what these skin cells, epithelial cells, are doing is in their cells, they are taking the DNA needed to make new skin cells, and they're making copies. They're just going at it, making copies and copies and copies, so they have enough for when they actually get into mitosis. That's all they're doing. So they are replicating, copying all of their DNA needed to make brand new skin cells. Once that phase is done, we get to G2, our second growth phase. In this growth phase, we more or less have them doing their normal thing, but they're prepping for their division. They're making sure they have everything set and ready to go. They are good with duplicating, dividing, etc. But other than that, they're still doing their normal thing. Similar to G1, but they have that added prepping for division. This is the end of interphase, G2. So interphase is G1, growth, S1, synthesis, G2, growth. That is our interphase, that is our interim phases. Then we finally get to mitosis, is the actual division of the cells. And there are 5 steps. We won't go into mitosis. We don't need to do that. We just need to know this phase because DNA replication, that's actually very important right now. So mitosis is the actual division of cells. That is what happens in mitosis. So that is the cell cycle. Now let's actually jump into specifically synthesis of DNA. That is very exciting. So within the cells, within the nucleus, you have all of your DNA. I think I might make a video just on synthesis of DNA because it's actually really cool. But we don't have to go into all those specifics. Just know that in the nucleus of the cell, that's where all of your DNA is, all of your instructions for all of the metabolic processes that need to happen, all of the functioning of the cell, what that cell needs to do. It has all of its instructions in the DNA in the nucleus of the cell. And how synthesis happens is within the nucleus and of the DNA, you have these proteins that go along and read each strand of DNA, and there will be a marker on the beginning strand of a specific segment of DNA that will say, I need to be expressed, or I do not need to be expressed. So to be expressed is to say, I need to be copied. So that protein reads it like, oh, I need to be expressed. It then goes along and makes a copy of the DNA, which is called transcription. Takes that copy to the doorways of the nucleus and hands it off to the messenger RNA. So the messenger RNA take that strand and translate it to actually turn it into proteins, etc. And that goes into more in depth. Okay, let's jump over to pyogenic granuloma and what all this has to do with pyogenic granuloma. It's pretty cool. Okay, so this article I have is specifically on pyogenic granuloma. So this one, I'll have a link in the description for this specific article. It's on uptodate.com, and this is a research article. I'm only able to read the first section of it. You have to have a paying subscription to read the rest of it, but thankfully, the very beginning tells us quite a lot. So since this is public record, anyone can read this, I am able to read it to you. So, I don't think I'll be reading word for word, but I have my notes to help me explain what I was reading, because again, this is some of the information that I had to Google to figure out what it actually meant. I suggest you all do the same when you are reading these. so you fully understand it, I do want to note, it says pyogenic granuloma, they refer to it as PG. Another word and more appropriate word or term name for this is lobular capillary hemangioma. So it's a benign vascular tumor of the skin or mucous membranes characterized by rapid growth and friable surface. Now, I looked up what friable meant, and friable, because in the vlog I openly said I don't know what that means. So friable surface is something that can tear easily, something that bleeds easily. You'll also see some definitions that talk about it can be reduced to powder very easily, but as far as with like skin and mucous membranes, it bleeds very easily. That is what friable means. And we already know in the vlog, I said that Ma's pyogenic granuloma, that growth, bled like crazy all the time. In this article, we are discussing pathogenesis, so the pathology and creation of pyogenic granulomas, which is what we want to know. Although the name suggests an infectious etiology, meaning if you were to look at the etiology or the meaning of pyogenic and granuloma, it would direct you to some bacterial infection, which we know it's a misnomer. That's not actually what causes a pyogenic granuloma, but the name stuck and they kept with it for some reason. Other than that, so far, no one really knows exactly what initially causes them to grow, but most theories on pathogenesis revolve around pyogenic granuloma as a hyperplastic neovascular response to an angiogenic stimulus with imbalance of promoters and inhibitors. So, hyperplastic, that was a word I didn't Google, but I'm going to assume hyperplastic, meaning it is highly active, as in it grows very quickly, the cells grow and divide very, very, very quickly. That's just what it does. Neovascular response, so neovascular meaning new blood vessels, vascular blood supply. So a neovascular response would be the creation of new blood vessels. It is responding to something. Something is causing it to make new blood vessels. And the angiogenic stimulus, angiogenic actually means the creation of new blood vessels. So a stimulus that specifically causes new blood vessels to form. And there's an imbalance of promoters and inhibitors. Now, promoters meaning it's going to promote this process. It's going to say, yes, make new blood vessels. You can do that. Inhibitors saying, nope, we actually don't need to do that right now. We don't need to make new blood vessels. Even though there's this stimulus happening, we don't need to do that. We know we're okay. So an imbalance would be, specifically in pyogenic granuloma, is you have a lot more promoters saying, yeah, do it, make more blood vessels, we need it, when really we don't. But the inhibitors are not functioning correctly or not responding correctly, and the promoters are having a heyday promoting the creation of new blood vessels. Now, these angiogenic growth factors. Now, growth factors, specifically, are naturally occurring substances that are capable of promoting cellular growth of blood vessels. So there are growth factors within our skin and cells that react to the angiogenic stimulus. So the stimulus that's saying, hey, we need new blood vessels, and this angiogenic growth factor is receiving that information and starting the process, saying, yep, let's actually do this. A specific growth factor is vascular endothelial growth factor, or VEGF, and decorin is a protein-coding gene, which is what's in synthesis phase of the cell cycle. Woo! So that is what decorin is. Transcription factors. So this is another angiogenic growth factor. But transcription factors are proteins that control the rate of transcription of genetic info from DNA to messenger RNA by binding to a specific sequence. Now, transcription factors. These are, if I remember correctly, I should have gone more in depth on this, but in the nucleus, on your DNA strands and segments, at the beginning of each segment, you have a protein that covers it, saying, nope, don't read me and transcribe me. I don't need to be read and duplicated. And then the factors can be off of it, or the protein can be off of it, saying, yes, read me. I'm ready to go to be transcribed and duplicated. The transcription factors are those. So those are the proteins that either cover the beginning segment or have it wide open. So we can assume with transcription factors, if there is an issue with the proteins on the DNA strands, depending on what phase we're at, for some reason, those proteins know when to express or open up certain DNA segments because they know it's time to synthesize all of our DNA. For some reason, they know how to do that on their own. I don't know how that happens, but with a pyogenic granuloma, something has gone crazy, and we can assume that those transcription factors are opening each segment, meaning they are off of each segment when they shouldn't be, and they're allowing the proteins to come in and read the segments when they shouldn't be. So this process is happening a lot more. They're making more DNA duplications for more cells during division. So this is happening too much. For some reason, those proteins are enjoying being off of the segments when they should not be. That's my assumption. So another angiogenic response or growth factor, along with the creation of blood vessels. As the blood supply, the new blood vessels are being created, we have the synthesis of DNA to make new cells for the skin. I'm going to assume what's going to happen is, since the body part, so the skin of my mom's finger, or whatever body part that might have an pyogenic granuloma, the response to the injury is to get rid of any infectious agent, the foreign object, etc. But it has to create new cells to fill in that injurious area and make it heal. So I'm going to assume that with that injurious tissue and the response to new blood vessels being created, the epithelial cells are also getting the message that, oh, new blood supply is coming in, we need to fix this area as well, let's make more skin cells. And this synthesis of DNA happens, and they quickly go into mitosis, passes over the growth, or it goes through that very quickly, back into synthesis again to make new skin cells, and this process continues to happen quickly, not at the normal rate, because we already know that an angiogenic response or a growth factor is the transcription factors covering or uncovering a segment of DNA. So proteins can read it when it shouldn't. Those proteins will continue to read the strands of DNA, make copies of it to make new skin cells and new proteins, etc. So this process is happening very quickly. It's not getting the message that, stop, we don't need to do this. We are okay. Stop duplicating and dividing. We're good. It's not getting that message. So it's going to continue to grow skin cells, epithelial cells, and more specifically, the blood vessels, which bring in that blood supply into the skin growth. And that's why it bled so quickly, or often, easily, etc. So that is my work through of this process and how it relates to the pyogenic granuloma. Now, since we don't know if there's an actual raspberry thorn or some other foreign object that got into the skin, we have no idea if that is the actual root cause of the pyogenic granuloma sprouting and growing, but we're going to assume that's what caused it, and that created a domino effect that was never ending until we chopped it off. So this cell cycle was going too fast, too often, and didn't know when to stop. The only way to do that was with chopping it off and then using the electrocautery to cauterize and destroy any remaining cells in that area that still had the message, we need to make more cells, we need to make more blood supply. Frying them, destroying them, killing them off so they stop. And then the surrounding tissue would go about its normal healing process in the correct way of making the new skin cells and stopping when that wound healed. So that is the pathology, the physiology of pyogenic granuloma, or what I think is the root cause, how it works, etc. Tomorrow we will be doing a hybrid of dissecting a sample op report of excision of a pyogenic granuloma and then coding it in CPT. So we're going to be combining the two into one instead of two separate videos. You can subscribe to this channel if you want more videos on A&P, ICD-10-CM, ICD-10-PCS, and CPT, and just my weekly vlogs of what we're doing this week. Subscribe to it, you'll get those notifications so you're always in the know. You can comment below if you found this topic interesting. Please comment below if I confused you at all, and I will go about explaining this in a better way, in a slower way, etc. I will help you out, but please let me know if there's anything confusing, if I rushed through it too quick, or if you wanted more visuals. And you can also like this video if you enjoyed the topic and me working through understanding some complicated information. It was a lot of fun. So I will see you all tomorrow in our hybrid dissecting an op report and CPT coding of a excision of a pyogenic granuloma. Bye!